Hi everyone, my name is Maude and I'm a PhD student with Yvonne Buckley in Trinity College Dublin. And over the last few years, I've been involved in a few projects that make use of PlantPopNet data or of PlantPopNet methods. So I decided that I would give you a talk today that would be some sort of a portfolio of Plantago projects. And that is that I will show in very few details quite a few projects. I will skim through some results from 4 different projects. And of course, I won't go into very many details about the methods of each of those projects. Well, I invite you to just use it as a conversation opener. We have the whole workshop. So if anyone needs or wants to know more, please contact me, of course. I'm happy to discuss any of these. I just preferred to open the conversation on several topics and have people who might be working on something similar hear about these today. Yes, so the first project I wanted to talk about is the size project, which might be known to a lot of you by now because there has been a manuscript circulating for a first round of comments a few months ago. The premise was kind of very easy. Size matters a lot for demographic processes. We know that in average, we can expect bigger individuals to, for instance, have higher reproductive outputs in many species in many cases, or higher survival probabilities. And on top of that, it's a basic information for many modeling techniques in demography. So I wanted to have a deep look into size. And the challenge when having a global data set, such as the PlantPopNet, well, there's many challenges, but one of them was, okay, so if I want to capture different populations in different parts of the world, the Plantagos there will face different environmental pressures. As a consequence, they might have different shapes. So how do I capture the need for a Plantago in a very cold climate to be, for instance, maybe very dense and close to the ground with dense rosette that will stay under the snow cover to resist frost in the winter, for instance, with something like what we might have in Cork, which is, so in Ireland, which is that Plantago lives with tall grass next to it, so it may need to make a few very thin, slender, tall leaves to catch out a little bit of sun despite a lot of competition. So how do I capture these sizes? So I decided to look for a metric that was measurable, that could perform well across rates and well across populations, and of course, that would meet the assumptions of any analysis I wanted to apply with it or to use it for. So I compared different size metrics from different studies that had already been performed and based on what we had from the PlantPopNet, and I performed my study at the GENET level. And I had several criteria. I will only show you one on this talk, which is here. So here what you have is on the y-axis, the R-square, marginal and conditional. So the percentage of explained variance for 4 vital rates, growth, probability of survival, probability of flowering, and reproductive effort, which is the number of stems produced, for the different size metrics as different symbols. And the 2 things that I would like to jump to your eyes from this picture are that they are not very far apart. We don't have one type of metric that really performs appallingly bad, and the metrics are performing pretty similarly in terms of of where they perform well and where they don't. And the number of leaf seems to perform fairly well for all the vital rates, bar a little bit of troubles with the reproductive effort. And the total leaf length was the second choice in the study. So number of leaves and total leaf length. So adding the number of, like the length of the longest leaves times the number of leaves, which is great because those 2 metrics are quite simple and they seem to perform well. So that was, to me, good news. So that's all I wanted to kind of skim through for this project. Another project I wanted to tell you about take place in the Alps. I was very interested when starting my PhD into investigating the link between demography and species distribution models or habitat suitability models. So in those models, one will make use of very readily available occurrence data for a species and predictors, environmental predictors that one has in the shape of maps. One will simply correlatively model the probability of occurrence for the species as a function of those environmental predictors. And given we have them in the shape of maps, we can then project this probability of occurrence, back into the geographic space. What we obtain is a map of probability of occurrence, which is often understood to be a map of suitability of the environment for the species. And these SDMs are really broadly used for conservation purposes, to assess the effect of climate change on species. Where will species be under climate change scenarios? But when using SDMs in an applied scenario, you sort of make the assumption that where species are, they are likely to persist, and that these mechanisms will stay the same under climate change. And these are assumptions on the field of demography, which rarely could be investigated. So there has been a couple of very nice papers, one stemming from our very own Anna Sergo, and Yvonne as lead author. But I was interested into this, and given that I knew that I would, a PhD is a long time, so I decided to also have part of the project where I would sample my own data, given, who knows, in a scientific career when you have 4 to 5 years in one spot. So I decided to make use of a steep suitability gradient for Plantago lanceolata in the Swiss Alps, because Plantago lanceolata is a lowland species, so it reaches its an environmental limit in this area. So I decided to model the suitability of the environment through an SDM and sample populations along the suitability gradient. So and when I say monitor the populations and sample the populations, I mean that I use the PlantPopNet methods and protocol, bar for the fact that I sampled them along the gradients. Part of my data is in the PlantPopNet network already, and most of it will come, or all of it should come into the network once I'm done with my PhD, just protected so that I can have my PhD out of it. Yes, so the SDM was performing very well. So this will only speak to the ones who know, but just skimming through, these are the evaluation metric of an SDM, so a species distribution model, and they are quite good. So in terms of predicting where the species is going to occur or not, the model is predicting fairly well. But now what did it mean in terms of population? So we built a population model for this setup in the Alps, which ends up being a matrix model in which we reproduce the life cycle that is shown here. I won't go into as much detail as this population model would deserve in this talk, because I'm skimming through several projects, but you're very welcome to ask any other questions. One particularity of the model is that more seeds produced by individuals will not necessarily create more recruits. We have a scenario where we would have a few microsites that get freed every year because there's a limited amount of resources, and only this many Plantagos will enter the population every year as recruits. And what will happen here is that individuals producing more seeds in any given year will simply be likelier to take part to contribute to the pool of recruits the next year. In terms of effective suitability on the vital rates, this here is showing you the different vital rates as rows. And what you see here are the parameters for suitability in the models. So if it's lower than 0, it means that there's a negative relationship. And if it's higher than 0, it means that it's a positive relationship. I won't go into every single bit of detail, but what jumps to the eye is that we don't have either all positive relationships or all negative relationships. We have different vital rates that do react to the suitability gradients in different ways. Sorry, having opposite reactions. And in terms of the result in the effect on the population, just very skimming through what happens if you iterate your population to a stable stage is that thanks to the constant recruitment, the population growth rate stabilized to 1 very quickly. What I do find is some sort of evidence for slower life cycles in harsher conditions. So I do have populations that behave in a different way, but the population growth rate is stable. That's all I want to skim through to just showcase what I've been up to. In terms of this Alpine project, I also was lucky to have a couple of undergraduate students who helped out gathering functional trait data from the setup. So I have a lot of information there. I was keen to find out that seed mass doesn't seem to vary along the suitability gradient a lot, nor through any gradient that we found, but the number of seeds actually quite steeply increases at higher suitability. So the number of seeds per inflorescence, which I found very interesting personally. The thing that I was keen on doing with those functional traits was to try to link them with the demographic, so the life history strategies that I found. So here on the y-axis, you've got, for instance, just as one example, the reproductive life expectancy, and which is one life history strategy metric for the populations. And here you have the mean SLA value for each population. And ultimately, when we use functional traits, we expect that they have an evolutionary meaning. We expect that they affect the fitness of individuals. And it's really rare that we have the demographic data to kind of make this in-between step between functional traits and fitness. So I was keen on to experiment with the data gathered by all the students and check what I could find. And what I found is here in this second panel here, which is that there is an interaction between environments. Like here, I simplified it by using just elevation, but there is apparently an interaction between climates and functional traits to explain the life history strategies. And it's a trend that comes back. And it's similar to what Ruth Kelly, who was working with Yvonne Buckley and published a nice paper this year. It's similar to what Ruth finds at the between species level. So quite interesting here. Then the last bit I wanted to showcase is more of a product. I was interested in also looking at the SDM at the global scale. So with a couple of colleagues, we've developed a few approaches to the problem, because obviously Plantago is it's very undersampled, as in we know that it's pretty much everywhere, and it doesn't have a very strong relationship to climate. So we have a couple of products here with different methods, and I tried to check whether what I was finding, for instance, of the tendency of a slower life cycle in harsher conditions would be maintained, at least in the native range. I do find that look at the confidence intervals on those things. It's sort of obvious. Not much is happening. If anything is happening, it is going the same trend as what I found in my only 700 square kilometer in the Alpine area. But not much is probably happening in terms of if you take into consideration the confidence interval. That's really skimming through, I'm sorry. One last bit is that obviously to get this life history strategy metrics for the native range, I had to develop some sort of population model, which I wanted to skim through, given that many people might be interested in this sort of things. At least that's the best platform I could imagine to show this. So I built this native part of the range of Plantago lanceolata matrix model in the same way that I built the Alpine one, with the same life cycle, which assumes that a fixed number of individuals enter the population every year. We tried several ways of modeling the recruitment, and that was the best. I also tried to explain this few vital rates here from the recruitment and the size of recruits through different environmental predictors, and nothing at whatever grid cell is available for the whole world was really being retained in those models. And then I guess it would require a whole talk just about it to go into the detail of how we built this matrix model. But basically, we parameterize it as an IPM. So that is going to be very nerdy, but I suppose there is a few nerds attending this talk as well. So we basically built it, we populated the matrix model just like we would build an IPM. So we considered that individuals could either survive and grow to contribute to the population the following year, or they could flower, produce seeds, which will contribute to a proportion of the recruits. And then the recruits will enter the population with a size distribution that is modeled by Model C. And all of these are mixed model, where we have the sites of origin and the year, the calendar year, and the plot width and quadrat that are included as random effects in the fixed effect model, in the mixed effect model. and the size where applicable. So really, that's what I wanted to say. It's quite basic. Anyone who wants to know more or comment more, you're very welcome to come and have a chat. Thank you very much for your attention, and thank you to all those who helped me a whole lot throughout these two projects, which are not finished yet. So anyone who has comments and ideas, I don't promise I will have the time, but I'm very welcome to hear and listen to any feedback. Thank you very much.